We are interested in the mechanisms for collective communication between neighboring and distant pluripotent stem cells during their early differentiation and cell pattern formation. We have chosen FGF-ARC pathway as primary focus since there is a lot of evidence in mouse blastocyst studies that the pathway induces the spatial organization during differentiation. However, the proposed FGF-ARC pathway needs to be evaluated in the context of human-induced pluripotent stem cells. To examine the pathway, we use small molecule inhibitors to block the pathway and see their effects in spatial context.
We are also developing a collection of Boolean network models for this pathway, which discretize the gene expression. The purpose of this modeling is to understand the priority of the mechanisms driving early differentiation. We also use our Boolean network models to inform an agent-based modeling approach which incorporates additional biological details like cell location. In this way, we are studying both the local dynamics and emergent behaviors associated to early differentiation and pattern formation.
Hello, this is Tejaswi Chanagiri presenting discrete methods for double-strand break DNA repair analysis of next-generation sequencing data. This is joint work with Margarita Ferrari of USF, Yonkyu Gion of Georgia Tech, myself, Natasha Janoska of USF, and Francesca Storici of Georgia Tech. We present a method for analyzing next-generation sequencing data obtained from a specific genome location where DNA double-strand breaks have been induced. The analysis is used to study the role of RNA in double-stranded break repair. Several experiments are performed under different treatment conditions, and the resulting DNA products are sequenced. From the sequencing data, we extract 20 nucleotide sequences around the break sites in order to study the variations produced by the RNA-mediated DNA repair under these treatments. Our approach is graph theoretical. Relationships among the observed sequences are displayed in a graph where vertices are sequences, and an edge between two vertices exists if the two sequences differ by a single nucleotide variation. By studying the structural properties of the graphs arising from the different experimental treatments, we hope to better understand the molecular processes that play a role in DNA break repair.
Hi, my name is Jonathan Bush, and my poster's title is A Torus Model for Optical Flow. A video recording, such as this one, is inherently a two-dimensional projection of a 3D world, and for the purpose of reconstructing the actual three-dimensional motion that was captured by the camera, it's important to understand the statistics of optical flow. Optical flow is simply the assignment of a sequence of vector fields, one per frame, so that every pixel is given a vector pointing to where that pixel appears to move in the subsequent frame. We propose a torus model for the space of high contrast optical flow patches, and we use tools from applied topology, including persistent homology and zigzag persistence, to validate this model. We also show that our torus model is naturally equipped with the structure of a fiber bundle, which is explained by the statistics of range image patches. This is based on joint work that was done by myself, Henry Adams, Brittany Carr, Lara Kassab, and Joshua Mirth.
My research is to look for evidence of compensatory mutational process in mitochondrial tRNAs. Delirious mutations can accumulate in natural populations, and reversal mutations are likely to be rare. However, new mutations at other sites can potentially compensate for bad mutations and reverse fitness of function. In a compensatory event, a single non-beneficial mutation in combination with another mutation to improve or restore the fitness. Mutations might precompensate later mutations and allow the genotype to move from one fitness peak to another one, or might compensate an earlier mutation by passing through a transitional state to restore the fitness. We are targeting tRNA because tRNA are essential, the low amino acids into the glowing polypeptide chain during protein synthesis. These genes are ancient and universal across the tree of life, and the tRNA molecular sequence have an extremely well-defined structure-function relationship. This allows us to draw some conclusions about fitness directly from sequences themselves. So, I am identifying patterns of compensatory evolution over different evolutionary timescales in xenoheptadous nematodes, including interactions between mitochondrial tRNAs and associated factors encoded in the nuclear genome.
Hi, I'm Young Kyu-jeon, a PhD candidate working in Dr. Storch's lab at Georgia Tech. This poster is about RNA-mediated DNA double-strand break repair in human cells. Margherita Ferrari and Tejasvi Chenagiri working in Dr. Jonoska lab have contributed for this work. Recently, many studies have been done to uncover the role of RNA in DNA double-strand break repair. In yeast cells, our lab showed that endogenous RNA transcripts could be used as a template for DNA double-strand break repair. But the direct role of RNA for DNA double-strand break repair is still unknown in human cells, such as whether the RNA can be a template for the repair or mediate the repair in a sequence-dependent manner. We developed a system in a plasmid to do a transient assay in human cells for studying RNA-mediated DNA double-strand break repair. We induced a double-strand break on the plasmid by using CRISPR-Cas9 system and captured the repaired DNA product by next-generation sequencing after inducing the break. We used our bioinformatic pipelines to calculate frequencies of double-strand break repair in different RNA sequence contexts and found that endogenous RNA transcripts can mediate double-strand break repair in a sequence-dependent manner through non-homologous joining, microhomology mediated joining, and RNA templated repair in human cells.
Hi everyone, my name is Nathan Rains, and on behalf of the Duke Tulane SCMB seed group, I'd like to present some of our recent results. We're interested in intracellular transport, which as you can see on the bottom left is a very complicated set of processes, but in particular we're looking at the end of the endocytic pathway with our late endosomes and lysosomes that are organelles responsible for the processing and degradation of cargoes that cells encounter in their normal environments. In order for that to occur, these organelles have to be trafficked across the cell to specific locations, and broadly we observe two types of motion for that to be facilitated. The first is motile, which as you can see in the green trajectory is very directed, and the second is stationary, which as it sounds means that the organelle is staying in one spot over time. And so to analyze our data, we employed a Bayesian change point algorithm to segment whole trajectories into pieces categorized by their individual states of motion. That is, we would break our trajectories into groupings by stationary, motile, and stationary as shown here. And then one notable result that we found was that organelles within the perinuclear region are much less motile than in the periphery. And that's notable because a lot of analyses that are in the field for this type of motion don't include region as a factor at all. So for our future work, we're very interested in delving further into the perinuclear region and trying to figure out what influences that drop in motility and assigning it to specific causes.
Intrinsically disordered proteins, ADPs, are known to be unstructured but biological functioned. They frequently contain various post-translational modification sites, PTM, and combinatorial PTM of ADPs is a prominent regulatory feature in protein biology. One good example is the N-terminal tail of G protein gamma subunit. It is intrinsically disordered, phosphorylation-dependent determinants of G protein signaling. Evidence showing that yeast G gamma subunit underwent multi-set phosphorylation events within its N-terminal IDR. These phosphorylation events modulated the activation rate and amplitude of the downstream mitogen-activated protein kinase. However, the role of intrinsic disorder in G protein signaling is still unclear. In this study, we engineered the sequence of N-terminal tail of G gamma subunit to have increasing order of structure. By this approach, we can see how well these mutants function as G protein gamma subunit in signaling process in yeast model.
Hi, my name is Boya Yang. I'm a mathematics PhD student at the University of Florida, the vice under Professor Li Binglong. I'm happy to attend this symposium presenting my research work titled A Unified Mathematical Model of Thyroid Hormone Regulation and the Implication for Personalized Treatment of Thyroid Disorders. Current clinician practice for thyroid hormone regulation of patients is based upon guesswork and experience rather than quantified analysis, which exposes the patients at a longer risk and discomfort. To quantitatively analyze the thyroid hormone regulation for patients of different thyroid states, we developed a two-dimensional mathematical model that can be applied to analyze the dynamic behaviors of thyroid hormones and with all result drug intervention. The unified model can be employed to study the regulation of thyroid-stimulating hormone and free thyroxine for normal thyroid subjects, such as monothyroiditis, and Graves' disease patients respectively. Given the early blood test data, we demonstrated the feasibility for healthcare professionals to apply our model in choosing an appropriate dosage regimen for patients to achieve the desired thyroid hormone levels within a specified time frame. If you are interested in the detail of the work, you can read the full paper by the link here. I will be happy to answer your questions. Thank you.
Pluripotent stem cells play a key role in embryogenesis, in that through their differentiation, the ectoderm, mesoderm, and endoderm germ layers are created. Thus, understanding when and how these cells choose to differentiate is an important problem in developmental biology. For this proposed project, we hope to tackle the question of when by creating a method that will find the time point where cells have organized into a particular germ layer, but not yet differentiated. We will take data on the cell positions in colonies that have been treated with different morphogens that feature in early embryonic development. Using this positional data, we will predict what particular treatment the colony was initially given. We will do this by implementing both a neural network and by processing the positional data through persistent homology. Thus, the end goal is to be able to predict what cells will compose what tissue layers before the cells actually differentiate into said tissues.